So I will give you now the first round of perspective in translation of the genetic code. Today we go from the amino acid, so the charged tRNA, and ready to enter the ribosome. And tomorrow we'll see the step of decoding the message at the ribosome level. You already got a significant amount of information today, but you will see from another perspective, in other details, and the principles of decoding, especially the proofreading step. How all that is based on experimental information. Here today also I will concentrate on the structural aspects, the way the structure influences the function. Of course, if you have questions, everything which I say is connected to function. Of course, it's important. And like the application about drug design are of some importance, why are these molecules, which are essential for cell survival, are or are not good targets for drugs? Like the question, where toxicity is the main. You don't want to hit these molecules, because if you hit these molecules, you just kill all the cells. So this is very essential. That was the problem tackled why you have antibiotics for bacterial and not human cells. You have to find the non-toxic things. Here is the same. So we can address this question at the end, why where synthetase is discarded from the target domains of drugs. And now they are entering in that target because you can see spots on the synthetase which hit essential function of the synthetase, but not the one which are conserved in eukaryote and human. Okay? So we can discuss that. So now I will stick to the basic chemistry of translation, and we'll follow the life. And one key player in all that transfer is transfer RNA, tRNA. That's why tRNA search you have seen yesterday going what is important, so I will go a bit faster. Just a glimpse on something I didn't show you yesterday. You just, when you look, you have the stems here, and when you assemble this part, the pocket here, with this part coming here close, you see that you have, I hope you see, you have insertion of bases, and these are the linker region. And these kind of interactions were used. I mean, tRNA was really the example structure, the first one, and the pieces of the whole tRNA, the block, were used as building block to understand more complex structures, like the ribosome was built on that knowledge, and then other. And now that, of course, with ribosome, there are much more information. But the pieces that you find there, you can find them also. So it's all assembly of blocks. So long. Another thing that I would like to stress before I go, and this question was addressed over and over, and it's a very important question. I'm going to show you data based on essentially crystal data, crystallographic data. And as Albert said before, these are frozen snapshots. But a frozen snapshot may be relevant in terms of function. Crystal is a solid state, yes. But how can you check that you are observing a functional aspect? One, for enzymes, like we are going to see, there is an easy way. If you can run the reaction in the crystal. If you run the reaction, you can charge in the crystal, you have a functional enzyme. Of course, the speed will be low. It will take hours, where in the cell it will take a short fraction of a second. That's a different story. It runs the reaction. So the trick that structural people use to have access to the function from the structural point of view, to the structure from the functional point of view, sorry, the other way around, is to freeze functional states. So you have trick. You will see tomorrow when you decode the messenger RNA, the idea of Gula Yusupova was to have the right sequence that could make the messenger pause. You know, it doesn't move quickly. So if it pauses, you have a stable conformation, and then you freeze it. Since the crystals are grown at 37 degrees for the ribosome, for example, and if you can freeze quickly enough, you can select a conformer. But the conformer is the 37 degrees, of course, freezer. For the synthetase, it's the same. Don't forget. So the crystallography is providing snapshots. And all the stories I'm telling you today, I will not enter on that, based on two facts. First, data of high resolution to reach the atomic model. Second, functionally relevant conformers. And sometimes you have artifacts, very often. You will see tomorrow when you come back to the decoding center, there is an artifact, because we're not looking at the functional things. I will come back tomorrow on that, just to listen, you will see. So you can create artifact. Artifact is not bad, because actually, as long as you can integrate that in interpretation, you see? So this is for the functional aspect and the technical aspect. If you crystallize, you don't mix, then just I get a crystal, I don't. I mean, this is what people have in mind very often. No, you have to infer and include our functional knowledge, your functional knowledge, to really grab the different snapshots. Okay, here we are going to follow the life of that transfer RNA. When the story began, we knew a lot about functional state, a lot, well, yes, a lot of things compared, but no information to the structure, and only good guesses about the enzymes which were carrying on the things. The transfer RNA, as Albert said, was predicted, its existence was predicted before it was found by Francis Crick. It's the adapter molecule. So it's a good example of things. It turned out to be right. And so here you have the information with the numbering. Albert mentioned, 1, 2, 3, coming from the 5' to 3'. This will be very important tomorrow when you decode the messenger, you see. These positions are not equivalent in terms of structure. Especially this one. Here you just follow a helical stack, and here you change. You have the wobble base. Okay? But here, normally, if you have a wobble type of base pairing, you are in trouble. This is the case, the near cognate position, where you have to select. For the transfer RNA, which will come back. So what you do, you have the anticodon where the message is, and the terminal, the CCA terminal end, the 3' end, which is the CCA, which is a terminal adenosine, where the aminoacyl is attached. So this is really the translation step of the genetic code. If you miss the boat here, you don't reach the right point. Because after that, the ribosome only cares about this part. Because it can have any amino acid, you can insert actually artificial amino acids. And that makes sense, because then what matters is the chemistry of the reaction here when you attach the peptide bar. And I just mentioned to you here that you have, at the beginning, an ambiguity, 2', 3' OH. So in terms of chemistry, whether you attach it here or here, it looks the same. It looks the same for most people, but not chemists. Here you have a sequence, another view. You see 3' attached. And things. So at this point, when the structure was started, the complex between this tRNA and the translator proteins, which are the aminoacyl tRNA synthetases, which main function, we'll see they have other functions, but we don't care about that today. I just mentioned that. They're very ancient. So genetic code and aminoacyl tRNA synthetase had to evolve together. And so the synthetase were had a duty to recognize the transfer RNA. The fidelity of that reaction is very high. It's one of the highest. I mean, you can understand why. Much higher than what happens in the ribosome. But at the ribosome, there are several steps. We'll come back later. But here we are on the order, 3 orders of magnitude, above 1,000. And there are a few problems we'll see. So we care about the fidelity. So we want to recognize the transfer RNA. And you realize that the codon here, the anticodon, which is essential for the transfer of the signal at the ribosome, is by no means essential for the synthetase. The synthetase can recognize the transfer RNA at any part. The only condition it has to recognize the correct transfer RNA for the amino acid. So you see the differences at the beginning. This was a bit overlooked. So for the synthetase, the anticodon is not essential. It is interesting because it's a nice part to be recognized, but it's not essential. And there are several isoacceptors that can be recognized by the same amino acid tRNA synthetase. That's an evidence. So we'll see the conditions of fidelity. But these are the questions. And to make a long story short, because this is already textbook information, you know, what kind of information did we have before the structure was available? Actually, I should come back. Was that, I told you, these two attaching groups were apparently the same, but on the ribosome, you have, this is the conformation. I mean, the attaching group, so the 3' end. But chemists were always, you know, a bit more skeptical on that. They were looking. And the group of Fritz Kramer in Göttingen, he was, you know, was a chemist, and then Alex Rich, they were asking the question, they were trying, before the structure of the complex was known, what is the primary target for the synthetase to attach, I mean, the ribosome? Because these are two different. How does it select? Do the synthetase always attach on 2' and 3'? And they had found that there were differences. So there is a chemistry involved in that, I don't go in. So there are some synthetases which were attaching here and some other at the end. And the 3' common target was becoming later when the EFTU would take over. At the ribosome, they were all 3'. But for the synthetase, you had 2' and 3'. That was a puzzle. OK, it was a table with a genetic code and 2', 3' relate to the synthetases, not only the synthetase. And then the investigation came out almost at the same time. But the first real interpretation came from sequence analysis, not structure. Sequence analysis, which allowed to compare the sequences of the synthetases. That was done, and I don't enter into the technique. This has been published already, and it's a textbook. And with a careful analysis of the sequences, you could divide the synthetase group enzymes in 2 groups, 2 classes, so to speak. And that was before the structure information was known, because the first structures were there is of one synthetase, and strangely enough, actually the first 2 synthetases, 2 or 3, I don't remember exactly, had the same active site, the so-called Rossmann fold, which is also called as nucleotide binding domain, found in other. It's one of the most, the first unit block found in structural biology was correlated to a function. Function was where. So the Rossmann fold was there, and so it made a lot of sense. You have enzymes doing the same thing, the same reaction, charging the transfer RNA. They must be the same class. They must have the same active site. This is also a good lesson for you guys. Never be sure. It's not because it's obvious that it is true. With these 2 classes, it was strange. You know, have 2 classes, and then just by sequence analysis, it was obvious that they were not the same at all, that you could not overlap. They were different enzymes. and you're in trouble. You know, just why do classes of enzyme to transfer the genetic information on the same molecules? And that's where the correlation between the 2', 3' occurred. You know, there was a table with 2', 3' there. And luckily enough, when you have the information of the partition, and you look at that, and it can happen, it did happen to me, to see, well, this is the correlation. One group is charging on 2', and the other one is charging on 3'. You say, well, that's a detail. I mean, what you can imagine, I mean, what is he talking about? You know, just 2', 3'. In life, chirality and asymmetry is essential. If you have a chiral center, 2 chiral centers, and then if I come back to that, this is a chiral center. This is another one. These, if you look at the environment, chemically speaking, these are 2 chiral centers which are different. And there is a strict rule in enzymology and chemistry. If an enzyme charges 1 chiral center, the active site is designed for 1 chiral, it doesn't work for the other. And you cannot change it with few mutations. It's much more than that. It's a basic division in life. That's a take-home message beyond just that. So, in fact, we were facing a very important division. And then the structure came almost, I mean, very same time. And these were the class 1, this class 2. These had the Rossmann fold. These had another active site, which at that time was very novel. Now there are very few. This is less common. These are the 2' and these are the 3'. When you have 2', 3', it means that the chemistry is a bit shaky. Cannot be either. It's one or the other. It cannot be both. Okay? But you can see the partition, and there are 20 amino acids basically, and you have 10 and 10. That also is a strange situation. I mean, I tell you because just think, when you find 20 amino acids for the message, and you find 2 groups of synthetase charging 1 and 2', 3', and you find that these 2 groups have equally distributed, 10 and 10, what is the reason for that? It makes no sense. Could have been, you could have 15 on 2' and 5, because after then, you know, it goes to 3'. It just tells you that there is a constraint and a message in the evolutionary scheme. There is a theory, I give you at the end a reference, there's no time, we have 40 minutes to give that, it's a totally different story. There are now interpretation of this partition. It's an evolution, you go from simple art, and once you split, you have to keep the symmetry. And so you reach equal distribution. It's very simple, and once you, like all the real explanations at the end, that's equal. So once you break, you have. And then another point, you will keep, and that's a message I want to convey because it's important everywhere. We'll see on the ribosome also. You look at the other difference. I told you about the active site here. I told you about the function. And now the tRNA. Let's transfer that. You see the images side by side. These are the active site. That's the anticodon. And you see that one synthetase, class 2, approaches the transfer RNA part. This is the key fraction from the major groove site. I told you, major, minor groove. And this one approaches from the minor groove site. That's why the CC has to bend to go in, while here it plugs in directly. This again, this is not trivial. It means this is a real important partition. Okay, you can oscillate within that, but you have an approach. And you will see when I come back at the end, if I don't have no time, just don't forget, you take that image. When the EFTU takes the synthetase, the tRNA, it's another protein, it grabs after that step. Take the tRNA, but the ribosome, it cannot go both sides. It can only be, is only one side has to be accessible to come close. Just these simple questions gives you key answer to understand the mechanism. That's why you cannot have. Imagine if the EFTU will arrive once on the protein side, another one tRNA side, you know, who would have to share it would be a very complicated system. And it arrives always the same way. position, move, everything fits well. It's like an engine where every piece is in the right place, and you cannot play around with the different place. Now we are going deeper in the situation. Just an example of distribution, class 1, class 2, with the transfer RNA. You see there are both sides. Of course, this is taken from today's structures. So this is just an image, a model that shows there is an overlapping. You cannot do that. But you can see that at the beginning you could imagine something primitive where you can charge either ways. But that's the position, 1 and 2. And then you charge. Let's go. What I told you about the chirality 2', 3', the enzyme, the active site. When you have the density, this is based on data, high-resolution data, so information, it's not interpretation. You have here the adenylate, and that's where the transfer RNA CCA end binds. The 3' class is on this side, and the 2' on this side. It's because the transfer RNA arrives. So you have always this asymmetry with a mirror image of that, very different in term. And that's key. Well, that's also in textbook, I think. So the fidelity of the reaction relies upon 2 recognition steps: the binding of the coordinate tRNA and binding of the correct amino acid. There are 2 steps, and you have to fulfill. So for the transfer RNA, we'll go very quickly. I told you, here you have an example of synthetase with a dimer with 2 transfer RNA, okay? And here are the active site and the anticodon recognition. But in other synthetase, the anticodon is not recognized at all. And what is important is to grab the correct transfer RNA. That's why on the transfer RNA, you have key positions which are so-called identity elements, positioned in different places. Here you have this position. These identity elements are important because the overall skeleton of the transfer RNA is kept, is conserved, and at key point you have the correct basis, and that is what the synthetase recognize. Actually, this was proven a long time ago by just flipping this residue on another transfer RNA, and then you will charge it with the synthetase. You don't have to switch all the transfer RNA. So it's like the testing selection process. For the correct amino acid, you have another problem. You have 20 amino acids, and you need about a minimum of 1 to 2,000 in terms of selection criteria in order to be safe on the introduction. And for here you have all the amino acids. For some of them, actually half of them, there are problems. What are the problems? It's that the stereochemistry of the amino acid is such that there is another amino acid or 2 others which are close enough to induce mischarging. That is, the synthetase will not discriminate the amino acid from the true one at a level sufficient for fidelity. And in that case, you are in trouble because you, for example, okay, the threonine tRNA synthetase that we'll look at between the serine and the threonine, here, sorry, I will go switch, the serine and the threonine, you see that the difference is just here. 1 methyl group. But it is obvious that if the synthetase is designed, the threonine tRNA synthetase, to grab this amino acid, this one will fit because it's smaller. Except the affinity will be a bit weaker, but 1 methyl group doesn't make a lot of affinity, which means that the mischarging, that is the rate of incorporation of mistake at that level, is about 1/200. It's not sufficient because that would be very dreadful for the fidelity of translation. That's why you have, for about half of the synthetase, an additional function, which is an editing function. So you know, all these enzymes are there to make the fidelity very correct. How am I doing? How much?
You have another 10 minutes.
Okay. So the synthetase here is an example of a very complicated one, where you have that's the synthetase active site. I mean, I take the threonine for one example, one of the, it's a class 2 enzyme. Here you have the active site where the CCA goes in and perform the reaction. It can bind the correct amino acid and two others, potential, valine and serine. But serine is the most dreadful in that case. So what happens, to make a short story out of this complicated event, is after the reaction is there, you have to check. So what happens, the transfer RNA, here the CCA moves from this place to this other module of the synthetase. And the synthetase are built out of modules. You have the catalytic site and then other structural modules, either for specific recognition or in this case, to check if the amino acid is correct. So it moves there and goes there and it is checked. So when you have these type of pictures, if that module is important for me, that's where the things occur. What you do, you make functional test. So you remove the N-terminal part, which is that domain. You just make a construct without the N-terminal domain, and you compare the wild type doesn't charge serine, but the one where you remove the N-terminal part charged, of course, threonine, slightly better, but also serine. So indeed, this is the domain which carries the reaction, the editing reaction. When it's not there, serine is charged about 3 times. I mean, not 10 times more. Okay, that is a control. The oscillation, trying to keep in the slide. So the mechanism, which was predicted a long time ago in terms of principle, and which is an enzymology principle. It's not unique to aminoacyl tRNA synthetase, but it was designed for discovered on aminoacyl tRNA synthetase by Alan Furst in the 70s. So there is a first selection where the catalytic site, where it's a first sieve, the chemistry selects. So you try to remove the methyl with the chemistry, and then the other one is the hydrolysis where you have the size, which means on the editing side, in this case, you play the opposite of what I said before. This side is designed for this serine. And of course, the good amino acid is being too big, it won't bind. It's the opposite of the other. The other will bind, but both bind, but one less than the other. And what is an enzymatic reaction? Very simple, like I said before. The substrate has to last long enough on the active side. It's a kinetic process and a thermodynamic process. If it stays there long enough, the reaction can occur and serine is released. If it doesn't bind or if it's ejected very quickly, no reaction. That's very general. So it's time thing. And that's how the selection occurs. And now we did more details, different structure. That's where I come into what I said at the beginning. This is the overall mechanism. Now, if you want to go at the molecular level, what do? You use analogs in order to stabilize different steps, and you freeze and you get the structures. So we are talking about very high resolution structures. You see less than 2 angstroms. I don't show you the details, but then you can see every atoms. And of course, when you have this here with 3 different structures, with 3 different substrates, and you compare them, you get the mechanism. You see that there is a water molecule bound with an essential amino acid. When you have then the structure, you understand, oh, why this histidine was already found very essential by genetic methods. Now we understand it holds the water, and the water attacks. Here, this lysine is essential, holds everything back. And then this water rings here, and then the other is released. The first water makes the break, and then everything moves away, and we go from here to here. water goes here, this one goes here, and you have disruption. So the picture of the three states gives you the mechanism and everything, and it works. Coming back, zoom, here is the general picture. Transfer RNA, reaction here, check here, and then the EFTU comes. You see that this part is accessible. Strange. That's where the EFTU are. It's not so strange. Class 1, I showed you an example of a class 2. Now there are other documented examples of class 1. The principles are the same. The stereochemistry is changing. But you see, here again, you have the editing domain, which is different. That's the case which was found in a leucine tRNA synthetase. And you see that there are two, the editing side, the synthetic sides. These are the two domains. But the same thing, you go from here to here. You have the translocation. So you run the reaction, and you check here the proofreading. So class 1 and class 2 have the same, except that you have always, you keep this mirror symmetry. In the class 1, it goes from here to here, and class 2 from here to here, because transfer RNA is the same. You see how this asymmetry is kept, this mirror image, you go all along. But at the end, on both sides, you have the EFTU, which comes. And, you will have, just to see, because these are all, the final step, I gave you the whole picture, the story, just to show you that there are novel data coming up. I gave you the basic principles. Now in details for the application and for every aminoacyl synthetase, there are a lot of interesting details. The mistranslation of synthetase has been recently reviewed with very recent result appeared last month. In Current Opinion Structural Biology, you will see the latest perspective with this point. And then one paper interesting also on the role of transfer RNA itself in the editing. But just to conclude on this part of the story, here is the EFTU. You saw yesterday, I showed you the EFTU story. Albert showed you another picture. Remember what I said? It binds here. And actually, it's not quite visible, but the rest of the protein is on the other side. So this side is accessible. And when you go on the ribosome, this is what goes on the ribosome. And this is the part which is positioned and goes on. And there again, we'll find that there are structural constraints, very simple rules. You know, they have to be positioned at the right place. There is a low resolution anchoring, and then a high resolution, very tight proofreading check through. And then everything is controlled by kinetics and conformational changes. They are both linked. So if you want a reaction to occur, I mean the ribozyme reaction, you know, you keep the right people in place for a long time, and then, well, long time. Fraction of a second, but longer than the wrong one. And then the reaction occurs. 5 minutes.